Hello everyone. Today I'm going to talk about a study based on in situ genetic correction of the sickle cell anemia mutation in the human induced pluripotent cells stem cells um, using ZFNs, also known as the zinc finger nucleases. This is a study published on NCBI in November 2011. So first we need to know about the disease and the reason we need to edit its genome. So the sickle cell disease is a group of genetic RBC disorder where the red blood cells become very thin and hard and often looks like a C-shaped sickle. These cells die premature because of lack of hemoglobin present in it. As a result, very few healthy RBCs live, which is known as the sickle cell anemia, and it can block the blood flow, which causes pain, also known as the sickle cell crisis. The symptoms of this disease are that the patient are very susceptible to infections and it causes a lot of pain and fatigue. There are treatments like medication and blood transfusion, but they are merely preventions and not lifetime cure. The only cure is the bone marrow transplantation and gene editing, but the transplantation of bone marrow is not done very often because they are too risky and can cost their lives. That's the only reason we need to edit genome in order to find cure. In India, there are less than 1 million cases per year, but in Africa, out of 1 out of every 13 black child are born with the sickle cell trait. Now coming to the basic working principle of ZFN, these are targetable DNA cleavage reagents which we use as genome editing tools. The ZFN induces the double strand bond to break and then the cellular DNA repair mechanism starts, which leads to both mutagenesis and as well as gene replacement at higher frequencies. The zinc fingers can be engineered to shortlist specific gene of interest in complex genomes with the help of its own DNA repair mechanisms. Thus, the ZFNs can be used precisely in higher organisms like humans. In today's presentation, we are going to study two methods, one with the help of the iPS cells and the other with the beta globin gene. So in the first method, we are simply going to infect the dermal fibroblast cells from two sickle cell anemia patient with the polycistronic stem CCA lentiviral reprogramming vector. Then some 10 to the power 5 fibroblasts are seeded in the MEF media. The MEF stands for the mouse embryonic fibroblasts, which are high quality feeder cells to nourish the growth of embryonic stem cells. Then after a week, these cells are then transferred to inactivated MEFs, and in a day the MEFs are then replaced with the human embryonic stem cell media, the HES media, and they are grown in 8 weeks until the colonies start to emerge. Induced pluripotent cells are picked manually and expanded again on the MEFs. The clones then gets transferred to feeder free culture in MTE SR1 medium after they are grown with some folds. According to the results, the DNA sequencing has revealed that the patient 1 has a sickle cell E6V mutation in one beta globin allele and a splice donor site mutation at the end of exon 1 in the other allele, while the second patient is homozygous for the E6V mutation. The infected fibroblast cells are then seeded in mitomycin C, treated with the feeder cells, and within a few weeks multiple colonies arise and gets expanded. Now we get to see on the left side of the slide there are some pictures. They are mainly the images of the face. They are mainly the phase contrast image of the iPS clones grown on the feeder-free cells, stained with immunofluorescence with the help of transcription factor like NANOG, monoclonal antibodies like SSEA3, SSEA4, TRA160, TRA181. The 4 lines of clones are selected for expansion of the iPS cells, 2 derived from patient 1 and the remaining 2 from the patient 2. These clones are then successfully propagated in the feeder-free conditions. These clones express multiple pluripotency markers at mRNA and protein levels, and they also showed telomerase activity like human H9 embryonic stem cells, which possess normal karyotype and can be differentiated in all 3 germ layers. Also, according to the image right beside it, we get to see that the iPS clone 1 and 2 both of them show similar pattern with the H9 stem cells, while the fibroblasts show no such telomerase activity in case of NP40. Now, in the second method, the zinc finger arrays which gets bind with the target sites are selected using the open method, also known as the oligomerized pool engineering. This is a method to engineer zinc finger arrays. Now, the selected zinc fingers are cloned on an XBA1 DMH1 fragment into ZFN expression expression vector that expresses the heterodimeric ZFNs fused by the T2A self-cleaving peptide. The CMV promoter then gets replaced with the EF1 alpha promoter. The plasmid also contain PGK promoter and GFP reporter gene, which can calculate the transfection efficiency. And the image beside here represents the beta globulin gene, including its 5 exon, the epsilon, and the gamma G and gamma R, delta and beta. The image beside it represents the open method. By this method, the ZFN selects a target DNA site and then choose appropriate pool to multiply. After that, they get selected with the help of a gradient of selective agents, and at last the arrays get selected. Now, coming to the results, using the ZFID web program, 2 target sites are found for which potential ZFNs can be engineered. These 2 sites are present 24 bps downstream as site number 1, and another at the other side of the mutation as site number 2, which is shown in the coming slide. So, the open selection of site 1 and site 2 are performed, and they have successfully yielded multiple zinc finger arrays. Now the quantitative testing reveals that most of them activates transcription by 3 times, which is efficient for function as ZFN in human cells. In this study, we used the ZFN technology as an efficient and stable correction of the E6V mutation in beta globin gene in independent iPS cells. The data shows that this technology allows efficient gene targeting of such mutations with no evidence for off-target cleavage in closed DNA sequences. The results show that reprogramming and ZFN induced gene editing can be used to create patient-derived iPS cells, and such kind of approach might be useful in future to develop therapies for monogenic diseases.